Welcome everybody to a really exciting edition of our webinar on emerging topics in biomolecular magnetic resonance. And I mean, we have two fantastic speakers. We will record, but I'm not sure if they have agreed to it. But anyway, I mean, we will then decide later whether we upload it to the ICMR-based YouTube channel. So just a reminder, please type in your questions into the Q&A box. And I mean, you can do this, of course, already during the talk, or you can raise your hand and then we can unmute you during the Q&A session. And I just want to point out, we always have this unofficial part afterwards, after the two talks. And it's very nice because one can have a nice discussion and ask further questions and so on. So please hang on and join us in the unofficial part. And there's early career researcher part of the ICMR-based webinar on magnetic resonance, so also please tune in there. With that, I want to stop my screen here and actually introduce the first speaker of today, which is Scott Prosser from the University of Toronto. He's a professor in chemistry and biochemistry. And I mean, just let me quickly tell you sort of his career path. So actually, he studied obtained a bachelor in math and physics at the University of Brunswick before he then joined Jim Davis at the University of Guelph to do a PhD in biophysics. He actually also spent some time in Germany. So in contrast to our second speaker, maybe he can still speak some German, which I'm not sure. And then basically he became, I mean, did some postdoc also with Gitte Wold sometime. And after that, he became an assistant professor and a professor then ultimately at the University of Toronto. And I mean, he's, I think, at least most recently, for sure, best known what he's talking today about. And this is really understanding the connection between structure, dynamics, and mechanism of action of GPCR proteins. So with that, I would like to hand over to Scott, just saying that, I mean, here one sees one disadvantage of the webinar. So it's not possible to go running with the speakers afterwards. So that's definitely a disadvantage. But so it's yours, Scott.
I just want to thank Marcus and the organizing committee for this chance to speak. It's great to see old NMR friends. Not that you're old, but I haven't seen you in a long time. And I'm also really looking forward to questions and discussions later. I spend most of my time these days attending pharmacology meetings and trying to think about the problems in the pharmacology community and how we can understand those problems, leverage those problems using NMR. And that's what I'd like to talk to you a lot about today. It's not going to be a traditional talk about a structure and a function, but rather looking at the pharmacology of GPCRs and asking what kind of NMR experiments can we do to really to bring our understanding of pharmacology to a new level. And that really involves marrying yourself to a whole new field of questions and thinking about NMR maybe in a little bit of a different way. And then I'd like to take a few minutes at the end of the talk to talk about something that we're all familiar with, and that is the chemical shift and the question of resolution. And even in a fluorine NMR experiment, is there some way that we can enhance sensitivity, enhance the chemical shift dispersion, enhance resolution of states by fluorine NMR? So I'll talk about some unpublished ideas that we've been working on in the lab, and I'm looking forward to sharing those with you. All right, let's see if the arrow key works here. So to start with, I give you a little bit of an introduction to GPCRs. There are over 800 G protein coupled receptors in the human proteome. So they're part of a very large superfamily that govern almost all chemical signaling. About 40% of all drugs target GPCR, so they're certainly very important pharmacologically. And in this cartoon, you see the GPCR depicted as sort of a potato. And you know, there are kind of two states. There's a cold potato state where the GPCR cannot engage this G protein, right? Which is this heterotrimeric G protein is the unit which gives rise to signaling inside the cell. So in the cold potato state, the G protein is suggested not to be able to engage the G protein. And then upon addition of an agonist, this potato switches to a hot potato state, it can then engage or bind the G protein, allowing for nucleotide exchange. And finally, upon pickup of GTP, dissociation of the alpha subunit from the beta gamma subunit. And as you notice in this cartoon, the alpha subunit then goes on to engage other proteins inside the cell. In this example, adenylyl cyclase. The beta gamma unit also gives rise to downstream signaling processes inside the cell. So it's a very exquisite kind of model for signaling. It happens over and over again in over 800 different GPCRs in the human body. And it all starts with an agonist at the top, giving rise to some kind of conformational change. So if you're an NMR spectroscopist and you wanted to try and validate this process, you could already formulate a few hypotheses. You could say, all right, I could implement NMR tags judiciously in certain sites in the receptor so that I could see the aporeceptor, the inactive state of the receptor, and then the active state of the receptor. But what is an active state? Because if you look at this cartoon, you appreciate that really activation involves a number of very discrete and unique steps. For example, precoupling, association with the G protein. And if this G protein is allosterically talking to the receptor, you might imagine that your NMR might be able to discern the difference between a nucleotide-free state and a GDB bound state. And so what you'd hope in the best of worlds is to implement some kind of NMR where you could actually chase this entire activation process and then give some sort of new insight into the dynamic processes. Now, the structural biology has really taken off in the last decade. We don't have hot potato, cold potato cartoons anymore of GPCRs. Rather, via cryo-electron microscopy and crystallography, we have these sorts of snapshots. And we have inactive, many high-resolution crystal structures of inactive states of the GPCR. And in addition, we have a number of high-resolution structures of the GPCR complexed with the G protein, both bound to GDP and also in the nucleotide-free state. So we can start to imagine with high resolution what the activation process looks like. And we can do more than that. So there are over 800 GPCRs. There are over 600 class A or family A GPCRs, where most of the pharmacology is focused. And so using computational biology and assessing multiple structures, we can kind of delve into the switches, the common switches within this GPCR scaffold that are involved in the activation process, that are involved in responding to the agonist and ultimately giving rise to some sort of cascade of events resulting in allosteric engagement of the G protein and activation. And of course, if you're an NMR spectroscopist, you would say to yourself, these would be very good sites to focus on to do the spectroscopy. Now, the reality for pharmacologists is a little bit different. A structural biologist would argue that it's all solved. And on the other hand, a pharmacologist would say, well, in fact, we know very little about the mechanism behind basal signaling. So basal signaling refers to the fact that the GPCR doesn't need an agonist. The potato doesn't need the agonist to convert to a hot potato. That many family A GPCRs actually have a significant degree of basal action. How does that happen? Then there's precoupling. So precoupling would mean that in the absence of a ligand, the G protein can actually engage or bind the GPCR. And then you have inverse agonism, which means that there's no signaling happening even in the presence of a complex. And then I could go on. There are some beautiful papers by Hoffman and Ernst from over a decade ago that talk about energetics of activation. And they suggest that the activation energy barriers are on the order of 20 kilocalories per mole. And somehow an agonist facilitates an activation process with these huge barriers in place. GPCRs are also largely promiscuous. So rather than having a single G protein, most GPCRs are, a good way to describe them would be soft. And a ligand can bias that GPCR to interact with a number of different G proteins or even non-canonical proteins. So case in point, the receptor that we study largely in our lab is called the adenosine A2A receptor. You see it depicted here. And you'll notice that at the C terminus, there's this large 100 residue plus disordered domain. And often this is truncated in the NMR experiments and the crystallography experiments, but it's part of the reality of this GPCR. And this GPCR, even though it's known to canonically signal through GS, will actually signal through GQ. And we know this from Brett's studies in our lab, and also GO. And that these pathways are dictated by the ligand. At the same time, arrestin is another common outcome of interaction with GPCRs. And then, of course, there's the interactome, which is vast. So in the case of the adenosine A2A itself, it can interact with other GPCRs or other proteins, some of which are suggested here are in the cytoskeleton. So you get the idea. It's far from a hot potato, cold potato picture. It's a very complex system, a very promiscuous system, a very soft system. It's also a system that, for that reason, is very challenging to do spectroscopy on. On the other hand, there are opportunities, there are questions that we as NMR spectroscopists can answer that have not been even posed, let alone answered. So the reality, though, is that NMR is, unlike optical spectroscopy and many other biophysical methods, NMR does require grotesque concentrations, to pull off this NMR. And so, if you wanted to study a GPCR in a physiologically mimetic system and at the same time incorporate a heterotrimeric G protein, you have quite a bit of of molecular biology to do in front. So in our lab, we use a bioreactor, and it's not uncommon to do 40-liter expressions in Pichia for 10 or 20 NMR samples. So we have this monstrous bioreactor with a 14-liter 14-liter volume. Then the scaffold proteins for making nanodiscs are made in E. coli, as is the alpha subunit. The beta gamma subunit of the heterotrimer is post-translationally modified, geranylated. And so one is forced to resort to insect cells in order to do this expression. But the beauty is that it can all be assembled and one can make a nanodisc, and these nanodiscs are remarkably stable. So we use a human unmutated adenosine A2A, and our NMR samples at 25 degrees will last easily for 2 weeks, 2 or 3 weeks. And of course, there are many other biophysical experiments you can do with one of these nanodiscs, but if you want to do NMR, you have to be prepared for large volume work. My postdoc Louis has the record in the lab. I think he's produced over 1 kilogram of cell pellet in some of his productions. So give you an idea where we're at with that. And so then what kind of NMR would you do? Well, there are lots of NMR, lots of methyltrosy, NH-trosy would be very sensible way of proceeding. On the other hand, if you think about it, many of these GPCRs, adenosine A2A included, have high resolution structures. And so we already know where the motifs are which move upon activation. So we can strategically insert cysteine mutants and then employ fluorine NMR in order to resolve inactive states and distinct activation intermediates. And this is what we do. And so in the lab, we have a host of mutants, but we focused more recently on TM5, TM6, and TM7. And the results that I'll talk about today are mostly on TM6. And the tag that we use to a cysteine mutant is a little bit exotic. You'll notice that it has a CF3 moiety. All right, so if you're doing fluorine NMR, that's important. It has a linker that it's a bromoacidanolide that allows us to link specifically to the cysteine. But then it has this intervening moiety, this phenyl moiety, and it's actually important. We've actually designed the tag such that this phenyl moiety is electron withdrawing, giving rise to greater chemical shift sensitivity of the reporter. And in fact, I'd like to dwell on some next generation tags at the end of this talk. So Marcus, if you'd be so kind, let me know when I have 5 minutes left, and I'll jump to the last part of the talk. And talk about ways that we can invent new tags that have far superior chemical shift resolution than what we can, currently use. So what's the difference between a nanodisc and a detergent? Well, the old detergents that we used were these maltose neopentyl glycol, MNG, commercially available detergents. They make the receptor very stable. You can distinguish inactive states and active states. These are fluorine NMR spectra, TM6 labeled, inactive on the left, active on the right. But you'll notice in going from gray to black, in going from detergent to to nanodisk, there's a huge change in the distribution, the resolution. So suffice it to say that nanodisk is a really important forward step. The use of phospholipids is a very important forward step in the study of these receptors. Not only that, it's next to impossible to study a GPCR complex with a G protein without using a nanodisk. So this is a bit of an overwhelming panel of fluorine NMR experiments where TM6 has been labeled, right? And in all cases, so red would represent different conditions where we add inverse agonist. Black would represent APO, that would be no drug. Blue would represent partial agonist, and green would be in the presence of full agonist. And in all of these spectra, you'll notice that there seems to be a bunch of peaks on the right and a bunch of peaks on the left. The peaks on the left correspond to an inactive state of TM6, where TM6 is actually parked in according to the crystal structure. And as TM6 moves out, the fluorine reporter becomes more solvent exposed, and as per usual, you see upfield chemical shifts resulting from activation. But in looking at all 12 spectra, what you can suddenly discern is that a number of resonances keep repeating. Namely, in terms of the activation states, there's no single activation state, but rather an A1 state, an A2 state, and an A3 state. And the relative population depends on the drug and the condition. And the conditions that we've used, in addition to drug, inverse agonist, no drug, partial agonist, full agonist, the conditions that we've used specifically involve the use of GDP-bound, nucleotide-bound heterotrimer and nucleotide-free heterotrimer. And you'll notice in many cases, there's a big difference between GDP-bound G protein and nucleotide-free. And you'd expect that because in one case, you're emphasizing more of a pre-coupled state. In the other case, you're now emphasizing a nucleotide-free activation state. So you'd expect to see amongst an active ensemble, a different distribution of states. And so we can take it slowly and just sort of go through some of these spectra. So this is the APO sample. And so first of all, sanity check, when you go from an APO sample to the, and now you complex this with a heterotrimer, you'll notice that the inactive state goes away. All right. And then you'll also notice that in the case of the APO sample, the addition of GDP-bound heterotrimer produces a strong upfield resonance. And it would make sense that that would correspond to a pre-coupled state. A pre-coupled state is a state in which the receptor is bound to the G protein, right? But it's not allosterically engaged, okay? And now it becomes allosterically engaged upon release of nucleotide, and you only see a small pickup of just a hint of really A1 here, because this is, of course, in the absence of an agonist. Now, inverse agonist would simply stabilize the inactive state, which we see. And on the other hand, you still do see active state representation, even though from functional assays, we know that there's zero signaling happening. So it's complex, but it's allosterically incompetent. Now, partial agonist, on the other hand, in this particular receptor, a partial agonist establishes a unique activation state upon addition of G protein and removal of the nucleotide. Full agonist is different. So you'll notice that in this panel, full agonist stabilizes the A1 state. So we have an explanation then, a conformational explanation for the difference in efficacy, which we see with partial agonist and full agonist. And the story, I can tell you that although this is unpublished, the story is even more interesting because GPCRs take advantage of what's in cell, what is in the cell, and what is in the cell, other adjuvants, cations, etc. And so there are additional distinctions between the partial agonist state and the full agonist state that arise from certain cations in the cell. And that's a story that I'll be happy to tell you at another time. And so that, you know, from this ensemble description, we can start to assemble a multi-state description of the receptor that helps us understand some of the pharmacology. And I didn't really get into the details, but in fact, we do see shifts in the inactive state that have to do with ionic lock disruption. So there are really two states that define the inactive state and three states that define the active state, two which relate to partial and full agonism, one which relates to pre-coupling. Okay. Now, if you have a nanodisk, this is a functional unit, so you can also observe G protein coupling. You could observe nucleotide exchange, or you could indeed observe hydrolysis of GTP. The alpha subunit is a GTPase. Therefore, if you feed it GTP, it'll churn that into GDP. And if you have a fluorescent reporter, which we do, there are commercial fluorescent reporters, you can actually register the hydrolysis of GTP. And that's what you see in this cartoon. So let me walk you through it. The blue curves have to do with this complex, heterotrimer, in the presence of GTP. And the magnitude of the blue curve has to do with, after a fixed point in time, the amount of GTP that is hydrolyzed. And so what you'd expect is that you'd expect that a full agonist like NECA, you would have the greatest degree of hydrolysis. Now, if you move into a partial agonist like LUF, that degree of hydrolysis is lower. If you have an aporeceptor, you'd have still a measurable amount, a basal amount of hydrolysis, but of course it's lower still. An inverse agonist would result in our hands, literally undetectable amounts of hydrolysis. And then finally, there are these orange bars, and the orange bars are kind of interesting. These are measures of hydrolysis as a function of ligand in the absence of the beta gamma subunit, just with the G alpha subunit alone. So it tells you that the beta gamma subunit in this cartoon shown here plays some kind of key allosteric role in facilitating the nucleotide exchange and hydrolysis. And we can chase that further with computational studies. So this is something I think that's really attractive for NMR people. If you have a high resolution structure, you can evaluate from a high resolution structure the effect of binding of an agonist. And what you would study, you could do MD simulations, but you can also do something that's more mathematical. You can evaluate changes in rigidity or local degrees of freedom in your molecule, right? And so that's exactly what's depicted here on this graph. Red would mean large changes in degrees of freedom, right? And blue or black would represent weak changes in degrees of freedom. And what you can discern from this type of analysis, rigidity analysis, are really changes in allostery. And what you really want to understand is, what does my allosteric network look like between where my agonist binds and where my nucleotide sits in the G protein? And you see something really cool. You see, well-known pathways that extend through the receptor, but then they also breach this N-terminal alpha helix from the G protein, and then this beta sheet region and portions of the beta propeller back to the nucleotide binding region. So what I'm trying to say is that via rigidity theory, we can actually confirm that the beta gamma subunit is playing a key allosteric role in facilitating the whole activation process. And if you're an NMR person, you should get excited by this, because you could think about NMR experiments that could validate these very models, since they have to do, after all, with changes in dynamics. So I don't know, Marcus, I think I'm burning up my time, so I'll just I go to an important message that I want to convey. And that is that the cartoon that I showed you at the beginning, cold potato, hot potato, is grossly wrong. And so one message that I would like to give you is that, in fact, to a large extent, the G protein is associated with the receptor, with the GPCR. And so to a large extent, the role of the drug is really to create or engage new allosteric pathways that facilitate signaling. So the changes in structure, in many cases between functional states, are subtle and small, right? But the dynamic consequences are profound. And I think that, we live actually in a very exciting era. This era of computational biology, crystallography, and cryo allows us to do serious NMR to do with dynamics. And so I'm actually really excited about the next decade. Now, I just want to change gears for a quick second. You people, my colleagues in NMR, are used to the idea of an NMR reporter being nothing but a nuclear spin. But in fluorine NMR, when you play with cysteine mutagenesis, your tag is bigger than that. So what I'm trying to illustrate for you is that you can think of a tag in fluorine NMR as consisting of a reporter region, an amplifier, which is really, really important. So in our case, we use this electron withdrawing phenyl group, and then a linker. And the question is, can we change this black box? Could we do better? Is there some way that we could in fact accentuate the chemical shift dispersion and therefore see even more? And so I want to show you some, well, one idea. So there are a class of molecules called pyridones, and pyridones do this fantastic Jekyll and Hyde tautomerization between a lactene and a lactam, and these are well known in chemistry. And it's also the case, these have been studied for a long time, the tautomerization is fast, right? So that if you were studying this process by NMR, you would see a single motionally average state in any given environment for lactene-lactam tautomerization. It's also the case that you can install a fluorine or a CF3 tag and a linker anywhere you want. So you can buy from good old Sigma fluorine or CF3 tags anywhere around this ring, 3, 4, 5, 6 position. And now the other point that you need to understand is that this equilibrium between lactam and lactene is affected by polarity. And so in a polar environment, so a highly aqueous environment, you find predominantly the lactam. In a hydrophobic environment, you find predominantly the lactene. But the question is, if you're studying proteins, would the microenvironment affect this equilibrium? And certainly, the microenvironment, as we know in proteins, varies considerably with regard to polarity, for example. So you'd anticipate a pretty sizable range of lactene-lactam equilibrium changes. And so again, these are the sorts of tags that are commercially available, no synthesis needed. There's a bit of synthesis needed to do the linking chemistry, but even that is not impossible because there are precursors that are commercially available. And so rather than do protein NMR, a good way of testing potential chemical shift sensitivity of a tag is in fact just to plop it in methanol or water or anything in between. And that's what you see here. You're looking at chemical shift changes of different tags as a function of polarity, going from pure methanol to pure water. And I want to point out some common tags on the right. This is a common tag that we used to use a decade ago, bromotrifluoroacetate. It's cysteine specific, it's small, right? This is the tag we use in our GPCR studies, this acid annelid with the phenyl moiety. And these pyridones you see decorated here. And in particular, draw your attention to this one, 4-fluoropyridone. This has a remarkable chemical shift dispersion as a function of change in polarity. And what does that actually mean? Well, if you change environment, these are some of the more conventional probes that you see at the top, and you can see the chemical shift changes that you see by fluorine NMR. Here's the tag that we're currently using. And these are some of these next generation tags, and you can see that the change is profound. And the beauty of these pyridones is the tautomerization is so fast that even in a protein, you would not anticipate to see some sort of exchange broadening. All you're doing is amplifying your chemical shift dispersion and thereby improving your sensitivity. This is unpublished, but it's an idea I'd love to chat with you, with people such as yourselves about. And just finally, I'll say that the nanodisk is just the beginning. If you have a functional heterotrimer and receptor, there are all sorts of biophysical studies that you can do and that we are doing. And so it makes for a really interesting biophysical chemistry project and allows you to get to the questions of pharmacology. And then one last comment. There are a lot of NMR things that I didn't talk about today. I think metal NMR is really interesting in GPCRs. I think ligand NMR is really interesting. We've been trying for 10 years to do biosynthetic labeling with compounds, with precursors that give rise ultimately to C13 labeled deuterated fluorinated N15 labeled tryptophan. And we've succeeded. And so I hope to share that with you at a future talk. And I'm going to actually skip my conclusions and just talk about my group. Looking for a new postdoc. The X's are people that have moved on. I haven't had a group photos since pre-COVID, but I should thank Kate in the middle and also Advait, who were doing, Kate has done all the receptor project stuff, and Advait has done a lot of the new probe design, the tautomer design, along with Jordi. Thank you very much for your time. It's really a pleasure to see everyone again.
Thank you, Scott, for a really exciting and inspiring talk. So we have already a number of questions. So the first one actually was raised or asked by Charles Saunders. And he's wondering if you could comment on your nanodisk. So do you use covalently circularized MSPs, and yeah, do you see any advantages in doing so? Or
we would love to use the circularized MSPs. It's just something we haven't gotten to. So we use the from the Wagner Hagen labs, the Delta 5 construct. Some of my colleagues in the GPCR world have said that the Delta 5 construct is a little bit too small for some of their cryo studies. And so I think a lot of the community uses either the 1D1 or the Delta 5 constructs. Delta 5 is an advantage in NMR because it's smaller. And the circularized would be even better. We haven't gotten to that yet. There are also some weird apolipoproteins out there that have cysteines in them that then cross-link and form even more stable units too. Those would be fun to investigate, but we haven't sort of dived into that. So we use the Delta 5.
Thank you. Then Jim Prescott asks regarding basically your protein and the native cysteines. I guess it's a more general question. So, do you have to mutate them out, or do they background, or are they tag specific, or how do you handle this?
There are a half dozen cysteines in that protein. They're all buried, and I believe 3 of them are cross-linked. And so we're lucky. You know, we have a protein that has a lot of native cysteines. They're all buried or cross-linked, and so we can add a cysteine mutant and do NMR with the addition of a cysteine mutant.
Okay. Then there's a question from Oleg Ansuntkin regarding your new tags, and especially sort of when you go from the CF3 to the 1 fluorine. So do you then have a 3-fold loss in sensitivity in this?
It's worse than that, yeah, because of course you have fast methyl rotation around the CF3 group. So you know, you're talking about losing 2/3 of your signal, and at the same time broadening your signal somewhat too. But the chemical shift sensitivity, the dispersions are significantly bigger, and we know this from a long time, right? People that do fluorotryptophan have seen in proteins chemical shift dispersions of 15 ppms. You know, so the dispersions are bigger with 4-fluoropyridone. They are bigger, 4 ppms in this specific study that we did. But on the other hand, so it depends really on if you can afford the signal to noise hit, you would resort to a monofluoropyridone or a monofluoroaromatic tag. If you can't, well, then you have to, for us, the 6-trifluoromethylpyridone still gave us in chemical shift dispersion that was bigger than our current tag. So it's still exciting. It was 25% bigger, but certainly true that the monofluoroaromatic tags are better.
Then there's a question from Geoffrey Bodenhausen. I mean, that follows kind of up this. So your 19F lines are rather broad, he comments, and can you distinguish between homo and inhomogeneous broadening? Is 19F relaxation driven by chemical shift anisotropy, and is it bi or non-exponential?
Yeah, so you're right, they're broad. I mean, on the other hand, the effective resolution in going from detergents to nanodiscs actually improved for us. So the overall dispersion became increased. So it's probably a consequence of dynamics. You're right, they're broad. I mean, our lines are 30 or 40 hertz wide, you know, and that's just a reality. On the other hand, with the BT-FMA tags, we can resolve what we want to resolve. There are some things that we see that you might not believe. For example, I'm showing here a deconvolution, 2-state deconvolution and a 3-state deconvolution. And you know, we believe that there's a peak here. This is the pre-coupled state, which under certain conditions is really, really broad and really, really dynamic. And then upon addition of GDP-bound G protein, you know, or agonist, etc., you see an enhancement in that resolution. And so there are a wide range of widths. And yes, we do rely on T2 measurements to make some of these claims. So we can certainly see differences between inhomogeneous broadening and homogeneous broadening. We're doing a CPMG study on a specific TM7 tag as I speak in the lab. Yeah, I'll leave it at that.
Yeah, thank you. So maybe take one more question before we move on. And this is from Genevieve Seabrook. And she apparently looked very closely at your slides, because on slide 11, she says, in the full agonist bottom spectrum, there's a peak at minus 61 ppm. And she's wondering what this is.
Geez, she knows her spectra better than I don't. Maybe we could talk offline about that, because off the top of my head, I'm not sure what she's referring to.
Yeah. Okay. So then, thank you again a lot, Scott. And we move on to the second talk. Thanks.
My pleasure.
Well, it's a pleasure for me to introduce the next speaker, whom I know very well for many years now, Óscar Millet. Óscar was a postdoc in my lab from 2000 to 2004. Óscar is trained as a chemist and a chemical engineer, and he holds a PhD in organic chemistry with Michael Pons. I first became aware of Óscar through his association with Art Palmer, where they published really a seminal paper in the field on the timescale of chemical exchange. Óscar is one of those guys who can pretty much do everything. I mentioned that he was trained as an organic chemist, yet in my lab, he was able to solve commutators of spin 1/2 spin systems for measuring relaxation properties of deuterons. And he did this essentially with no instruction from me. He simply read Art's book, and that's specifically on spin 1/2 particles, and then went off and did it himself for spin 1. He's worked on studying energy landscapes of proteins via dynamics and scalar couplings. He's determined the energetic cost of domain reorientation in sugar binding proteins. And then since leaving my lab in 2004, he's focused his efforts on a number of a very diverse array of topics, including allostery, protein dynamics, protein electrostatics, and now metabolomics. Speaking of Óscar as one of those guys who can pretty much do everything, I should also add that he's a superb chef. I've had the opportunity to verify that on a number of occasions. He's also an extremely good tour guide, and he took me and my son all around Bilbao to a number of very interesting beaches that we enjoyed. So Óscar is really one of those people who makes having a lab and going through all of those hassles that we all have to do as PIs well worthwhile. So it's great to see you again, Óscar, and the floor is yours.
Thank you very much, Luis, and thank you everybody for the opportunity of sharing the work with you today here. And I have no problem acknowledging that I feel completely embarrassed by the fact of being introduced by a giant in the science as Luis it is. So for which I really appreciate, Luis, you being around to just introduce to me. So I will discuss a little bit of the things that we do in the laboratory. First, I have an introductory slide on the recent things that we do. Recent means in the last years. We've always been, as Luis was saying, kind of dispersed more than diverse, probably, but still there is a kind of conducting, you know, like meaning on the kind of research that we do. And certainly I'm interested in protein stability. And the first topic that I will talk today here is about trying to find some pharmacological chaperones for congenital erythropoietic porphyria. I have to apologize because not much NMR will be shown here. I think it's an interesting topic. NMR is there. I'll try to highlight it as much as possible, but this is a very broad project that encompasses many different techniques, as you will see. The other interests in the lab are related to metabolism and metabolomics. Metabolomics that has been covered here by different very, very interesting talks, and I will skip this part because for the sake of time, and also because it's been covered extensively. And I will introduce a recent application to the description of metabolism using phosphorus NMR in my second part of the talk. So talking about porphyria, essentially, it's porphyria is actually a rare disease that it's actually found in people that have an impairment in heme production. So it's a disease, it's a metabolic disease completely related to a misfunction in any of the enzymes that account for the heme pathway, to the point that actually the specific enzyme that is actually ill functioning will correspond to a specific number of porphyria, that they share many common aspects, but they also have their own particularities. So we became interested in congenital erythropoietic porphyria that is associated to a misfunction on uroporphyrinogen 3 syntase, which is the fourth enzyme in the pathway. Porphyria is a very severe disease, particularly this specific porphyria is really, really grave. And the poor souls that are actually undergoing this disease really suffer a lot from plethora of symptomatology that you can see on the right side of the slide. And particularly, there are many characteristic symptoms like porphyrinuria, means red urine, that can be helping in the diagnose, hemolytic anemia, of course, and many others that I will be discussing later on. So essentially, because it's a rare disease, and this is partially something that it's kind of beautiful, because in many diseases it's very difficult to establish what it would be a proper genotype-phenotype relationship, or what it's normally called in pharmacology, that the target, the protein that you target is properly validated. But in this case, it's the case. I mean, the protein, clearly the mutation in the protein produces the disease, is what you see here. This is actually what is called autosomal recessive. It means that you have to inherit a mutation from both progenitors, and indeed the severity of the disease is proportional to the kind of mutation that you inherit. And among the frequency, most of the mutations are pretty much equally distributed, but there is one mutation, C73R, that is particularly frequent in all the patients. And unfortunately, this is also one of the mutations that is most severe from the phenotype. Of course, this is what we call the genetic defect. The genetic defect is unfortunately accompanied by a metabolic defect. The metabolic defect means that in a healthy individual, remember that we were talking about heme production in the pathway. So this enzyme, represented here by the arrows, means that it catalyzes the reaction from pre-uroporphyrinogen up to uroporphyrinogen 3, and this goes on to heme and so on. But even in healthy individuals, we have a small amounts of uroporphyrinogen 1 also being created there. Actually, in the urine test, is currently tested commonly in all the urine tests. The thing is that in a healthy enzyme, normally this is running normally, and your amounts are very small. The problem is that when you have a CEP patient, this porphyria, obviously Uro3S is ill functioning, the activity is low, the production of uroporphyrinogen 3 is impaired, and the pre-uroporphyrinogen actually accumulates as in the form of the porphyrin, the toxic porphyrins of the type 1, uroporphyrin 1, that it evolves in certain reactions, but it doesn't go off. This is represented here as this accumulation in this basin. Essentially, this creates many problems. This is what it's called the metabolic defect, and most of the symptomatology specific for this porphyria are attributed to this accumulation of these porphyrins. Why? Because those porphyrins are actually photosensitive at the visible light, not the UV light. It means that those patients cannot really go out to the street because they get burned, literally. They also get accumulated in teeth or in the eyes, and those porphyrins are obviously fluorescent, and they increase the risk of infection because of the skin sensitization, and in the end, it produces blistering and deformations that produce the ulceration, laceration, and the loss of certain organs. The body reacts producing some defense mechanism, like for instance, some hair, and this is called hypertrichosis. So most of this symptomatology is associated not with the heme defect, but also with the accumulation of porphyrins. So a long time ago, we started in the lab to trying to understand, originally by reasons that have nothing to do with the biomedical problem, but increasingly devoted to it, to trying to understand what was really going on with this enzyme that could become so pathogenic. And it seems that those enzymes have a common feature, and this is what they call the stability defect. The stability defect means that the enzyme, even though looks like the folded conformation is actually stable over time, it is not. Like most of the proteins, and particularly in the heme pathway, at least uroporphyrinogen 3 syntase, is indeed kinetically stabilized, which means that the protein is active, and then what you see here is the secondary structure measured by circular diacharism, a simple experiment, just measure over time in the open circles. And you can see that just simply this protein is continuously degrading towards like a final state that is not there. It's precipitating essentially. And the closed circles correspond to the enzymatic activity that essentially mimics the amount of protein that you have. So essentially, two conclusions can be drawn from this very simple experiment. The first one is that the protein is not stable. And the second one is that this instability is the cause of the loss of functionality that you find in the protein. And well, we analyzed that by cryo-electron microscopy to show that this was actually deaggregated. So this means that in real enzymes, we actually don't really deal with proteins that are in thermodynamic equilibrium. But this is fine because evolution doesn't shape proteins to be stable forever. This only needs to be the protein stable for the homeostatic time that is needed. And this is enough for the wild-type protein. I mean, look at the numbers, it's 3 days almost. So it's plenty for exerting its function in a normal cell. The problem arises when you look at the mutations, because when we analyze the mutations, the enhancement factor here represents the number of times faster that the protein degrades as compared to wild type. And then you find that some of them really accelerate its degradation rate. Surprisingly or not, C73R, remember that mutation that was the most common one, accelerated to 400 times. And this is really into the true time scale, into the perturbing the cell functional activity. At this level, though, in the in vitro experiments, all we see is that those mutations, and you can see here in the chromatograms where we separate between the porphyrins, the healthy porphyrins would be the type 3 and the type 1. And you can see in the dashed line would be the wild type behavior. And then you can see that the mutations produce two effects. One of them is the reduction on the uroporphyrin 3 production as expected because the enzyme is being degraded, and consequently the increase in the uroporphyrin 1 accumulation because the spontaneous degradation of the substrate. So that is in vitro. Then what happens in the cell? How this instability or stability defect translates into the cell? Well, it's what it's called homeostasis. Like essentially lack of stability means lack of stationary concentration of the protein, and this has been described by many people many times. And this is what we can see here in this experiment. In here we express GFP versions of the different mutant proteins, and you can see that wild-type protein accumulates while the mutants do not accumulate because they don't show fluorescence because there is not enough concentration in the cells to produce that because of this impaired homeostasis now. So this stability defect is actually functionally relevant for the cell and presumably also in the individual. So if the problem is stability, perhaps the solution is what it's called pharmacological chaperones. Like in the canonical definition, what you find there, pharmacological chaperones are actually molecules, low molecular weight, normally synthetic compounds that either induce correct folding by binding to the protein, increases trafficking to cellular destination, or increases the intracellular activity on the enzyme. I mean, this is kind of a wish thing more. Essentially, in our definition, we think molecules that actually bind to the protein and increase its residence time in the cell. And that's why, that's the definition that we use in order to look for those molecules. In order to do so, with GS, we have developed a drug discovery platform. And this is a complicated slide. It doesn't really matter. It means that we use, as many people do, many of the different tools that are currently used for drug discovery. But essentially, we have personalized that to pharmacological chaperones. And we do three things. The first thing that we do is actually do two different kinds of tests on a very large number of molecules. It means on the thousands that are screened. One of them is actually a thermodynamic test. It means that we check normally, we check whether it binds or not, and whether this binding is actually stabilizing in terms of thermodynamic with the protein. And this is what you see here. This actually tells you a lot about the true binding, but it doesn't tell you whether it will work on the cell. And in parallel with the same number of molecules, what we do is do an homeostatic test, which means that exactly the same as we did before, we have one of these destabilizing mutants that don't show the fluorescence and then try to see whether the fluorescence is recovered in the presence of the compound. This is fine. This is a functional assay, but doesn't tell you whether this is by the action of direct binding. So we cross the results from the two assays, and then we get a short list. And then this short list is actually characterized extensively by NMR spectroscopy. And then that's what we believe is actually our main singularity. And I must confess that I added this slide as a tribute to Luis, because his legacy on methyl-trosi, it's been, I think, instrumental for many of us. And then we also use that extensively in the lab, even though I won't show you that here. And this is just a beautiful example that we recently showed in an almost 100 kilodalton protein that we also, is not the case we are using for this project, but another project that we're working on, pharmacological chaperones, where you find a dimer of 100 kilodalton, where you can see very beautiful spectrum. And incidentally here, like just by adding certain compounds, one of them you can see in the insets there, that one of them differentiated between two different signals. This actually was interpreted that it was binding and desymmetrizing the dimeric structure, and allowed us to expand the chemical entity of the molecule just by analyzing these spectra and increasing more than tenfold the affinity for the interaction with this protein. So with a long time, like a long effort with this platform, we found for this target, for the uroporphyrogen 3 syntase, that we found a molecule that actually is a repurposed molecule. It's called thiclopirox, which is an off-patent antimicrobial agent that it's normally approved and off-label now for topical and fungal infections, but that according to our experiments, our results indicate that it's also a pharmacological chaperone to stabilize Uro3S, and that's what I will show you next. Essentially, this is the structural model used by NMR spectroscopy. Normal chemical shift perturbation, uroporphyrogen 3 syntase is a 30 kilodalton enzyme that can be analyzed by a regular HSQC. And then we can use Haddock in order to get a proper model with the chemical shift perturbation on the binding of the compound. More importantly, we demonstrate as well that the molecule actually has kinetic stabilization, and this means that we can stop this kind of degradation of the protein in a reductionist approach, in an in vitro approach, as you can see here with a deleterious protein, where it actually degrades, and then when you add PX, it partially recovers the kinetic stability. This kinetic stability is translated into an increase in the activity, not because the enzyme is more active, but just because the enzyme is active during more time. It's the concept of time-dependent activity. And you can see here that indeed you can recover pretty much in vitro the activity of the enzyme as if it was well-typed during a while. That said, then we have to go to animal models. I know this may be far beyond, but actually this is essential in drug discovery. So you have to have reliable animal models. In this case, the genotype, phenotype expression, this relationship, which is largely bijective, allows you to obtain very good animal models. Unfortunately, and just by introducing a mutation on the genome of the animal, you can pretty much observe the same kind of features that you observe in the phenotype of a patient, like red color urine, as I said, a large accumulation of porphyrins, as is shown here in the chromatogram that we obtain in the lab routinely, or the porphyrin accumulation in bone, in eyes, and also the skin photosensitivity. So this is actually a good animal model. And then when we test our compound during 45 days in our lab with the animal model, we see two kinds of things here in the porphyrin. First, you can see that the porphyrins go up as expected, like this corresponds to a more than 2-year time in a human being, Whereas with the CPX, the cyclopirox treated, we observe a reduction between 35 and 40% of the porphyrins, the toxic porphyrins in the serum in blood. Importantly, we also see that proton 9 levels go up. Proton 9, as you can see here, they are actually related to heme. We cannot measure heme directly, but we can measure proton 9. And this is actually indicating that the carbons are using properly in order to produce what it would be the appropriate heme pathway. So essentially, with this result, I mean, this actually is also translated into the physiology of the organ, like the splenomegaly, which means that the inflammation of the spleen is heavily reduced by the action of the drug. The spleen is related to the processing of the drug, of the heme, of the erythrocytes that are non-productive, and therefore an inflammation of the spleen corresponds to what is called erythropoiesis. So the inflammation of that means a return to the normal pathway in the spleen. And we also can see kind of a reversion of most of the features that you can observe in histology. So there are different stainings in histology. I don't pretend you to see them, but you can see in the arrows that, like, for instance, there are white spots in the first of the histology. This corresponds to fat, essentially, which is called steatosis in medical terms. This is mostly eliminated. This is like ham, you know, the more reddish, the better. So like this is what you see in the liver of the treated compounds. In the second one, you see the iron accumulates that are actually reduced. And we also observe other markers for fibrosis and, most important, for inflammation as well, which are consistent with the reduction in the spleen size. With all these results, essentially, we apply it to the FDA and the European Medicinal Agency regulatory, and then we obtain what is called the orphan drug designation, which is the first status for developing this drug. We are essentially actively developing the drug further, and we are now in many other aspects. I can discuss that in the questions if you have interest on that, but we are really pursuing the drug development because we believe that this is actually working nicely. Now, that said, I mean, I put on hold that because I will show you that actually this is not completely unrelated to what I just showed you. I wanted to share with you a second piece of work that we recently developed, mostly Ganeco Bernardo in our laboratory. And this is actually a phosphorus NMR analysis of liver metabolism. So essentially, because we are interested in metabolism, we have a problem with that. And the problem we found is that the metabolism is overly complex. You have too much information, and sometimes too much information is as good as too few information. And when I look at the metabolism, like, for instance, that you can analyze with several platforms, or you could even imagine device where to choose from, my feeling is that I feel like in Tokyo. So essentially, I cannot really drive through. Like, it's really too complex because there are too many pathways and too many information. So it is desirable to obtain a reduced yet comprehensive view of the metabolism. This is all obviously impossible, but our approach is actually try to understand what it would be the phosphorylated moiety of the metabolism. And this has several advantages. First, the pathways that are actually phosphorylated are not important because everything is important in the cell, but they are related to many fundamental functions of the cell, such as the energy, the glucolysis, gluconeogenesis, oxidative phosphorylation, and also, for instance, to the integrity of the cell in the phospholipids. Therefore, like, this is a little bit more like the Bilbao subway. I mean, Bilbao is a smaller city. There I can manage properly. So essentially, the idea is that try to simplify the metabolism. We are measuring the phosphorylated metabolites. This, of course, it's not new. I mean, we don't claim that we are the first one at all, nor the best ones measuring the phosphorus metabolism. I mean, actually, phospholipid in the liver was, like in terms of like the molecular description of medicine was probably at the early stages, even in 1909, was described as the essence of fat. And there have been many works in biological phosphorus, both in tissues, and particularly in MRI techniques, and we don't claim anything of that. There are but like the thing is that when we faced literature, we saw that there was still something that it could be done. And first, we tried to adapt some of the existing methodology to try to see whether we could really measure with that the liver metabolism. And then we took some liver from mouse, try to, starting with some protocols that were described more or less and see what happens. And then we faced like significant problems. And then the first problem is, so we have to really look at them carefully, even at the level, for instance, that in the simple mixture that you had at the beginning in chloroform and methanol, we have to analyze that in terms of with even the phase, because sometimes we would get biphasic extraction, and sometimes we would get a monophasic extraction. And this was probably because the water coming from the liver tissue was not properly and not constantly identified. And this was because in the treatment one that was described in the literature, this was too close to the interface between monophasic and biphasic extraction. Then we take another method in the literature, and then this was really close insight into the biphasic extraction. But this was there was another problem there, that was that the solubility was fractionated, and some of the compounds could have partial solubility in each of the fractions. And in the end, we had to really work at and try to optimize accounting for all these effects. And this is only an example. What they try to say is that in the end, we came out with an optimized protocol, applying all these novel techniques in terms of chemometrics and metabolomics that really know how to work in reproducible conditions in order to obtain reliable data. That includes standardized operating protocols, and includes also, for instance, adding some, gadolinium agents, as it was in the old times. Obviously, people would do it, but also Franz Mulder and Claudio Lucchina beautifully described for the use in metabolomics recently. We use essentially the recipe that works nicely, and all these additions were not in the literature. So in the end, we came out with an optimized protocol to measure both phases. And with that, we could determine, and this was another addition, up to 50 different metabolites that could be quantified in an absolute way and in a very reproducible way with more or less 5% error. Those metabolites essentially, in order for the assignment, we had to do spiking with more than 200 referent compounds, and it was extremely useful. We started using the colloq experiment, the phosphorus proton colloq, but in the end, what it was really useful was to use a long-range HHQ-MBC toxi experiment that allowed us to obtain the assignment of many of the compounds, a unequivocal assignment. Of course, one of the problems of phosphorus is actually the chemical shift sensitivity. It's also sensible to many aspects, osmolarity and so on. But now that we have measured many of these liver tissues in different projects, so we can also get a library of the chemical shift variations. And now we are working in a program that actually will do the automatic fitting, taking into account all this variability that can also be accounted for. So with that, this is just my last slides to show you an application, and then I go back again to the project of the porphyria, because then we said, okay, so we have the livers from the porphyria mouse and the porphyria mouse treated with cyclopirox, and then perhaps we can have a look to them. And then our strategy is always combined with histology. And this is, I know it's something different, but it's our approach. I mean, we are always at the edge of the biomedical application. And then we believe that what it's useful this phosphorus analysis for is to add the molecular dimension to what it's normally in the medical field analyzed histologically alone. And that's what we did with these mice. And then you can see here, for instance, in the lipophilic fraction, how you see that the spectrum of the set mice as compared to the wild-type mice is heavily distorted in a wrong way. Some lipids are upregulated. And this upregulation of the lipids is what you see histologically in the fibrosis and in the steatosis. The steatosis can be seen in the oil red as these red spots there, whereas the fibrosis are these white strips in the serous red that you can see there. But this is beautifully shown as well in the lipids compositions, for instance, in the triglycerides or total cholesterol and the PC and PE ratio. I told you, and not all of them. I mean, we also added a few of the protonated, non-phosphorylated metabolites, 4 or 5 of them, to complete the metabolic analysis. When we add the cyclopyrox, though, our surprise was that actually the levels of the lipids and all these lipids were recovered back to almost normal, and this was manifested as well in the overlap of the spectra. And this adds beautifully, and it's very meaningful for us to try to add a molecular dimension to what we analyzed previously histologically. Another thing that we could look now in the soluble fraction was several markers that we have identified as apoptosis, for instance, ADPR, and in a lesser extent, some nucleotides. Those are actually prompted us that in CEP mouse, there should be some apoptosis. And we had no idea, nobody described that before. And then we had the histology down, and then we could look, and then here the apoptosis you can see into different histological analyses. One of them is for caspase 3, and the other is for PARP. What is called, there are 2 markers of histology, and then you can see these brownish markers there in the histology, where it is true there is more apoptosis. I can understand why, because cells are actually ill, and then the apoptosis is upregulated, but this was not described, and this was an outcome of our phosphoromic analysis. And then again, when we add the cyclopyrox, things tend to be normal, probably because of the regulation of the pathway. The liver is easily reversible, and in 45 days, there is enough time to go back to normality, and then all this apoptosis is actually gone again, as it is also shown in the metabolic marker by phosphorus NMR. So essentially, that was pretty much it. And I only have a take-home message. There is NMR and other rigorous biophysical approach. In my case, mostly, learned that in Toronto and in Barcelona. Combines well with cell biology and even physiology. I know that this can be look like the extreme ends of science, but to me they are not. And then they can be, why not, merge to investigate timely biomedical problems. And this last but the most important slide is actually the acknowledgements. This work, like the porphyria work, has been done by many people in the lab during many years. And I wanted to acknowledge to them all. The phosphoromic analysis is mostly the work of Ganeco Bernardo and John Bilbao, with the collaboration of many other people in the lab. But this is their extensive development. Also, I would like to thank some collaborators. We extensively work with the French group in Bordeaux for the porphyria, and more recently with also the Mount Sinai School of Medicine with Bob Technik. And that's it. Thank you very much for your attention.
Okay, thanks very much, Oscar, for really demonstrating how NMR can liaison with other techniques that at least some of us in the audience might not think about too much. Chuck Sunders has a question. He wants to know if you have a series of kinetic and cellular stability measurements for your mutants, your CEP mutants.
Oh yeah. We have we actually, for all of them, we calculated the activation energy by measuring essentially you measure the temperature dependence of the kinetic rates. We measure that at the beginning with circular diagram. They fit normally well to an Arrhenius model, and then you can get the activation energy, which would be the enthalpy towards this transition. Is the transition state.
Okay.
And so they are not that. I mean, all the mutants reduce extensively this barrier.
And maybe I misunderstood or didn't quite catch it, but the enzyme seems to be pretty small, that particular heme enzyme, right? It's like 30 kilodaltons or so that you showed a spectrum of, or?
Well, no, the spectrum I showed, and this was 100 kilodalton. Like, but this was a different spectrum. This I just show it to say that I also do methyltrosy, Luis.
Oh, okay. So that was just to cover your bases.
Like the other one, I didn't need to do methyltrosy because we could assign the enzyme. It's 30 kilodaltons. It's a a bi-domain protein.
Right.
Yeah.
So that's my point. Sorry. Was there a comment?
Please go ahead.
Anyways, that was my point. If it's only 30 kilodaltons, it seems to me like you could do a fair bit of NMR on it per se. No, I mean you did it to determine this thing that bound this little tiny chaperone. But I'm wondering if you could establish whether you had a whole plethora of different mutations that decrease the stability of this enzyme. Is that merely because you're throwing darts on a board and wherever they land has an effect, or is there some sort of global mechanistic effect that's responsible for the decrease in stability?
No, of course. We tried. The problem is that the mutants don't last long. I mean, even for wild type, it's difficult because it's this kinetic stability. So essentially the samples, I mean, at the concentrations of NMR, because this is concentration dependent, those kind of aggregation processes, obviously, at the concentrations suitable for NMR, generally your proteins don't last long and they are not that nice. I mean, the wild type is nice enough, and then we assign the protein. We have the purely assign the protein. We use that extensively for drug discovery, for other non-repurposed compounds. And then we know essentially how it works. And then we also try to investigate a little bit more on the mechanism, because it's a weird mechanism of the enzymology. But with the mutants, you can do little because they just disappear from the spectrum. Then when we analyze that by cryo-EM, it looks like either swap domain or just simple aggregation of kind of folded monomers, but essentially it's a solubility problem, kind of like, they keep stacking together, and this is kind of a general mechanism.
Luis, can I ask a question?
Sure. Sorry, Art.
So you might have said this and I missed it. When you showed the structural model of the compound binding to the protein, was that some kind of allosteric site that stabilizes the protein, or does the compound bind reversibly to the active site, stabilize the protein, but it comes off long enough to let the protein be active?
No, that's an allosteric site, far from the active site. And this is actually, we call it the allo site. This has an advantage, which is that it does not interfere with the active site. And it has another advantage, which is not mutant dependent. So essentially, it is not a site where any of the pathogenic mutations particularly is. Actually, we have data that shows that it can improve the amount of protein in the wild type enzyme as well, which can also be exploited for certain applications. Yeah.
In the screen, then, did you have some way of biasing against compounds that bound to the active site?
Yeah. We do a trick. I don't know. It's probably published, but we just thought about that. So you do a million, you do a docking with 1 million compounds, and then you count them. So the docking is probably bad, but the first docking, you use that to detect the spots where it binds. So there are hot spots in the protein where it will bind. So what we found is that pretty much 50% of the molecules will bind on the binding side. Okay, that's what you would expect, because the binding site is kind of shaped by evolution to bind things. But about 17% of the molecules would bind in a place that actually was, out of the blue, you don't think about that. And if you dock many molecules, you find those things, because you explore the entire chemical diversity, and then what you're looking for is actually for the protein component. And with that, we found the allo site.
Just to follow up, how good are these docking programs? I mean, did you verify with the NMR study that you've done? Could you verify that the compound got it?
The pure docking, I to your standards, awful. But Haddock is very good. Once you add the NMR chemical shifts, if you got enough, in principle, you can get a good model. Yeah.
Okay.
But then again, even with the limitations of Haddocking, for instance, for the application that I just mentioned, it's pretty beautiful because you pretty much average out things, So like and on a second layer, a kind of a comparing thing, then for that is useful. But I wouldn't believe a model that it was not supported with experimental data just by docking. And actually, when we try that, very often what happens is that they sorted up by energies. Like if you try them as a beginning to do that, those energies are totally meaningless.
There is
I would also maybe have one, if I may. So, I mean, I remember this. I mean, this chemical chaperone for transthyretin. And I mean, of course, transthyretin, if it's not stable, it forms amyloid. So does this protein also form amyloid-like deposits in patients?
No, it's not amyloids. Like it's folded aggregates, which is another, I thought it was amyloids. That's why I had a look to it. But transthyretin, it has a very, very weird thermodynamics because if I recall properly, the unfolded state is actually more stable than the folded state. And it's completely kinetically stabilized. I think I may be wrong, but I think it is. And then the final state is even more stable than both. And this is amyloidogenic. In there, there is little you can do, and there you don't have chaos. In this case, you don't have chaos either. So once it's gone, it's gone. In the cell, it's homeostasis the same. But the problem is that it doesn't even go through the unfolded state. Here it's just drains out from the folded state directly, which is it's a more common way of aggregation that you would expect.
Thank you.
There's an additional question in the Q&A.
Sure.
Could you comment on the C to R mutant? Is the structure known? Does it form a salt bridge interaction, perhaps? And also in apoptosis, which protein does the compound bind?
No, the C73R, there is little you can do because the half-life time is less than 15 minutes. So essentially, we have some spectra that essentially only shows already nothing. And in the circular dichroism even, it really decays very, very quickly. That's why we have a different. So what we did, I don't have the data to show, but this is the C73 is a hotspot for stability. So what we did is actually mutate to the different side chains on the amino acids, and then what you find there is that the bulk, the volume of the side chain is related to the stability in there. So it must be something related to kind of in this particular mutant, it probably means local unfolding at the local level in there. And it won't be aggregation unfolded, completely folded protein for sure. But it's very difficult to work with this mutation.
Even if you throw in the chaperones to try to stabilize it long enough to...
Yes, the chaperones do stabilize that, but again, the chaperone doesn't work that well. In the end, and this is something that discussing with David Rubinstein, I was not aware at the beginning, but you stabilize a little bit, but a 3% stabilization in vitro would correspond approximately to a 10% homeostatic stabilization in the cells. And what we do is a very tiny effect. So we are not really, I mean, you stabilize things and there, but this means that a lot of equivalents almost working at a more biophysical than biochemical level. So but the reason why we believe it works is just because there is a very wide range of activities that are acceptable for this disease. For instance, the C73R mutant, it has only a 0.5% activity, yet the organism is viable. So it means that you go from 100% activity to something that is 200 times less activity, and still you can do something. So if you improve perhaps to a 3% activity, there is a huge improvement phenotypically speaking. But biophysically speaking, it's only 3%. You know, it's
but we're on the edge anyway, so any little improvement can make a huge difference. And I would imagine that in terms of the proteasome system, having the proteins last a little bit longer means you have a lot less buildup at the level.
That is the case, and then we published that, and for reasons that we will understand, I have to say that we published that in PNAS, and the inhibition of the proteasome works nicely, but it cannot be used as a therapy because those patients are chronic. So it can be a proteasome inhibition. You could imagine that it works for certain cancer treatments where there are, temporary uses, but I could foresee toxicity at many levels, but it does work. Like you can see also phenotypic improvement by inhibition of the proteasome with either bortezomib or MG132.